podocytes are like a heart of the kidney's function. So our lab is interested to understand why these podocytes first of all undergoes injury, why they become get damaged. People with anemia, because low iron levels, if I try to give the drug that can induce the hypoxic condition, he can cure it. That was that for that approach, he got 1/3 of Nobel Prize actually. But he referred my paper, which we published later, how these developmental origins of adult diabetic kidney disease. That is actually theme of my lab. We love to protect the kidneys, and we try to offer at least some therapeutic solution to the mankind to prevent the podocyte injury and podocyte loss. I'm dreaming we come up with some strategic therapeutic options very soon. That was in my optimistic approach. Yeah, and I mean it for that.
Hello sir, how are you?
I'm good, thanks. So I hope how are you then?
I'm good. I'm good.
Sorry for long ride for you.
No problem, sir. So you work on diabetes and in context of functioning of kidney cells. So can you elaborate on that? What is the hypothesis? What is the problem that you are solving? And also how you are doing it in lab?
Okay. See, every organ has its important function. Similarly, kidney's main job is to two things actually. Kidney maintains the homeostasis. It regulates the fluid balance. It regulates electrolyte balance. It regulates acid-base balance. Besides these things, kidney's another important function is it prevents the loss of proteins into the urine. Our blood is a soup of proteins. It could be immunoglobulins, it could be carrier proteins, it could be transfer proteins, and several enzymes and hormones for that matter. They're all are bathing in the blood, and they go to different organs to serve their function. So this is a kidney act like a sieve. Sieve means it will only allow the small molecules pass through. Bigger one will retain back in the blood. For example, small molecules such as excess glucose. When in the summer, kidneys will reabsorb the water, but in the winter, kidneys allow the water to go through in the urine.
Yeah.
So like that, but they never allow.
That is the reason why we pee a lot in in
right.
winter.
So they never allow the large molecules to go in the urine. So kidneys homeostasis, I give an example. In summer, we don't, even though you drink a lot of water, we don't urinate much because kidney reabsorbs, because kidney senses the temperature is very outside is very high, so it will preserve the water. Yes. And in the winter, it knows that you don't need much water, so it will let the water. In the winter and rainy season, you go more urination, even though you drink very less water, because kidneys don't reabsorb the water. They let the water to go through in the urine. So kidney's important job is to filter in. Retain the large molecules such as importantly proteins, filter out the small molecules, and especially urea, small amino acids, they all pass through the urine. So to do this function, it has a sieve. For example, a typical flour mill is there. Now flour, how the kitchen, our parents or mother will sieve out small flour will be small molecules of the grain will pass through, large will return back. So there large ones are good ones in the kitchen, but here also large ones are good for us.
Okay.
They have to be there. So kidney's job of filtration is entirely depend upon a barrier that is called kidney barrier or glomerular filtration barrier. Glomerulus is a small tuft of the tuft in the nephron. For example, kidney is in the big picture. Kidney is a big home if you consider. Nephron is your the front hall. And within the front hall, it's a glomerulus is a important place of your house, very important where everything is happening. For example, your hall or something kind of thing. So now, when you zoom the glomerular further, it has as I said, glomerular filtration barrier. It has a 3 layers. The filter has a 3 layers. Bottommost layer is called endothelium, which is coming along the blood vessels. Of course, endothelium is common across the body, but endothelium is also one of the part of this barrier. Second one is membrane. It is called glomerular basement membrane. Third one, they are podocytes. So podocytes are upper, interior is a endothelial, in the middle membrane, basement membrane. So now podocytes, as a name said, podocytes, they have the finger-like projections, like food process. So what they do, if this is suppose, imagine this, my hand is a blood vessel. Inside the blood vessel, halo, blood completely filters, sorry, blood flows. So now there is a basement membrane enwrapping this one. Now podocytes completely provide a epithelial sheet, like, suppose if you have a, imagine if I have broke my hand, they'll put the, like, what do you call,
Plaster.
plaster of Paris, this like the wrapping, completely wrapping.
Yeah.
So podocytes completely wrap
Okay.
this blood vessel, and podocytes contribute for the components of the glomerular basement membrane, and also it will provide the growth factors that are essential for endothelial cells.
Okay.
So altogether, in a nutshell, podocytes represents the entire filtration function of the kidney.
Okay.
So they're crucial.
Okay.
And these podocytes have a, suppose if you see my fingers, you can't see anything between the fingers. So these finger-like projections are wrapping the glomerulus, blood vessels. Glomerulus is nothing but tiny blood vessels. Podocytes are wrapping the blood vessels with these podocytes,
Okay.
which are called extensions of the cell. This podocyte food process is known as extensions of the cell, cytoplasmic extension of a cell. So they're covered like close together, they're tethered like this. Now what happens with aging, with the diabetes, of course in specific conditions such as HIV nephropathy, in case of extreme conditions of exposure to the hypoxic conditions, so many several disease conditions, the gap between these food process becomes slightly bigger.
Okay.
Now protein that is passing through the blood vessels can escape from the blood and go in the urine. That condition is known as proteinuria.
Okay.
So now, as you heard, that podocytes are like a heart of the kidney's function. So our lab is interested to understand why these podocytes first of all undergoes injury, why they get damaged, why they detach from the what do you call membrane, and why they appear in the urine. And in some disease conditions like diabetes, obesity, HIV nephropathy, for example, even COVID also. COVID virus actually lives in that virus. Coronavirus lives in the HIV virus. They live in these podocytes.
Okay.
So, so our crux of our work, the crux of research interest in our laboratory is to understand how these podocytes gets damaged first. Is there any possibility to recover them, to slow down or to stop those damaging events, and to maintain the healthy glomerular function, so as to retain the kidney function? That is the main thing. So what are approaches? If you see the etiological factors for this kidney damage, especially in diabetic people, people presented with excess levels of the glucose, excess levels of glucose derived compounds called as advanced glycation end products. Because glucose, excess glucose, they directly reacts with different proteins of the blood or circulation. So glucose has a highly reactive nature is there because of its aldehyde group. So proteins which have a what do you call positive charged amino acids are there, lysine and arginine, histidine. This positively charged amino acid have an extra positive group, NH3 plus group, that is very prone to react with the aldehyde group of the glucose or fructose for instance, C double bond O, keto group of the fructose. They react and form advanced glycation end products. So they all cause damage to the different cells, especially podocytes also. Excess glucose has one problem. And in the diabetic people, especially type 1 diabetes people, they are presented with excess growth hormone. It is a different, various hormones upregulate in diabetes. For example, diabetes is presented with low insulin. When insulin is low, some hormones like a glucagon, growth hormone, cortisol, they go up. So we'll see how these the hormones that elevate in diabetes conditions have a threat to podocytes.
Okay.
See, anything what we eat, if at all it is absorbed into the blood, anything what we drink, it is absorbed into the blood, that must and should go to the kidney. Because anything that enters into the circulation, suppose what we eat, it will go into the, if not digested, it will go to the large intestine.
Yeah.
That is a fecal matter. If anything goes to the blood, that should go to the circulation, and that all molecules will expose it to the different, podocytes will be exposed to all these molecules. Suppose we ate drugs as a medicines, they all go to the kidney. So podocytes are under constant exposure to these agents, good or bad or ugly. So they have to face this consequences of excess any molecule that enter into your blood circulation.
Okay, now I can correlate why drinking alcohol is damaging your kidney when you are saying that it is actually damaging the podocytes. And if the podocytes are damaged, then the filtration process will not happen, and
Yes,
that
yes,
will
yes.
result in all the phenotypes that we observe of a disease. So, like, what actually you are doing in lab? Like, how you study these podocytes? What is the
Yeah,
process?
actually, for to study any disease, we need a model.
Mhm.
A model system helps us to get more insights,
Mhm.
and also a model system helps us to correlate with a really human situation. So, as I said before, podocytes are located very deep inside.
Yeah.
Um, Harvesting podocytes from animal is even model animals like rat and what do you call mice.
Mhm.
It is a bit challenging thing. So I have a cell culture model. Like we have a human podocytes. I'll explain in a minute how we got them. Then we also have this model system called obesity related. To understand the obesity related complications of the glomerulus, we have a obese model. And third one, we induce diabetes in mice by giving a drug called streptozotocin.
Mhm.
Streptozotocin is a drug when you administer it into the mice or rat, it will damage the beta cells of the pancreas. So animals will develop the diabetes in 48 hours of administration. So that after, if you maintain this streptozotocin induced diabetic mice or rat for a 4 weeks of time, they almost resemble the human diabetic situation.
Okay.
So then we can study, like we can dissect out the kidney, we can study the kidney function in the intact animal.
Mhm.
That's one of the aspect. So coming to these podocytes, we have cells. These are the podocytes isolated from the children
Mhm.
in United Kingdom.
Mhm.
So what happened was during my when I got this job in India, I went to UK. Is my collaborator's name is Professor Moin Salim. He's a renowned nephrologist and is both MD, PhD. Is a scientist as well as physician from the University of Bristol. So I worked with him. So then what happened? I liked this model. Then I requested and obtained all permissions. I brought those cells to India.
Mhm.
So probably in India, myself and there is Anil Vasudevan. His name is also Anil. He is from Saint Joseph College, Bangalore. He also worked with Moin Salim. So
Okay.
both of us have these cells. So so what these cells, very tricky thing. These cells are actually to maintain these are primary cells, am I right? So they're not cancerous cells or cell lines.
Mhm.
So what the Professor Moin Salim did was normally they proliferate actually at 33 degrees centigrade they proliferate. So he introduced a thermosensitive antigen. What happens when you shifted these animals to 37? That means our body temperature. They start differentiate.
Okay.
So they after 12 to 13 days of differentiation, they really resembles podocytes that actually exist in a adult human being. So you have to maintain almost 15 days those cells, every batch of cells to do any experiments.
So perfect model system to study
Yeah, as of now,
podocytes.
this is the only model, cell culture model that is available across the globe. And one more thing, these podocytes also called as salimocytes after Moin Salim. His parents are actually from India, but
Okay.
they migrated to UK after independence. So I hosted his talk. Actually, his mother requested me, Anil. I did PhD. She did her MBBS, and both his father and mother, Salim's father and mother, did MBBS at Usmania General Hospital.
Okay.
So as a nostalgia, whole family of Salim visited. We had a nice talk in 2019 at Usmania Medical College. So I'm thankful to him for donating those cells generously, because it's very difficult to get permissions from Medical Council of UK, Bristol, University of Bristol permission, University of Hyderabad permission, DBT and all. With a lot of risk, I got these cells, and we are very helpful. They're very helpful, and I am also helping a lot of students here. I supplied these cells to many
Okay.
institutions in India. So these are our model system. Mice, one thing is cells like podocytes, and third thing we approach physicians after proper permissions. Like for example, doctors at Osmania General Hospital, there is a Professor Manisha Sahai. She is my collaborator. Whenever they wanted to study biopsy of a patient on other purpose, if she through proper channel, proper permission, she gives me, she shows me, she exchanges with me the pathophysiology of those patients, human patients. So I learn from them. So
So
these are
what those cells are telling you?
oh, these are sections actually. We don't
Okay.
get cells from the patients, but we
Okay.
can isolate the cells from both animal and we have in vitro cell culture models. So we study those cells, and we see the physiology of those cells or structure of those cells by electron microscope.
Okay, electron
Yeah,
microscope. I mean, so you study healthy cells and affected cells and then compare
compare.
and
Yeah,
try to understand what is going wrong in the affected cells. So
excellent,
how
excellent.
that process is
Yeah,
going on?
you asked very nice question, Mustafa. Uh, Healthy cells, you always need a control. But
Mhm.
I will tell about what we study actually, and what kind of pathophysiological conditions I'll tell. Very challenging thing. I'll tell a couple of things here. In some states of India, especially from Uddanam region of the Andhra Pradesh, and the fluoride contamination areas of the different parts of the country, and our neighboring country Sri Lanka also has this problem. This is endemic kidney disease.
Mhm.
So people found that some pesticides or some silica that exists in the soil coming through water, it causes the nephrotoxicity. So what we do, we'll take some amount of silica or pesticides, we treated these cells. Of course, we slightly use the excess concentration than what exactly there in the like water, because to mimic, because we have to show the results little faster. Otherwise, cells we cannot maintain to that age of the human being. So we do excess concentration to just understand the pathophysiology. So what are different disease conditions what we mimic in our laboratory. First of all, a CKD, of unknown origin, like pesticides, whether they cause nephrotoxicity or podocytopathy.
Mhm.
Again, just a silica. Then hypoxia. Indian military, I have a defense grant actually. I had a defense grant. So DRDO has a unique problem. Suppose you are a healthy person applying for a job in
Mhm.
a defense, and I am already imagine I am serving as a lower cadre in a defense, and I want to apply for higher cadre or career advancement scheme. When they do physiological testing, your kidney function is very much normal, and my kidney already started dysfunctioning. So, see, the person who is already working in a defense, for example, they are working in a Siachen area or high altitude, their kidneys already started dysfunctioning. So there are some Tibetan population have high proteinuria. So high altitude hypoxia is major factor. We also mimic how hypoxia is toxic to the kidney or not.
Okay.
Yeah, and
So is
then
it toxic?
yeah, hypoxia is not good actually. Hypoxia, kidney is the first organ that tend to react against the hypoxia, because kidney produce erythropoietin.
Okay.
See, for example, you are a mountaineer.
Mhm.
After crossing the physiological, what do you call, the sea level, your body senses little hypoxia, because you little
Cannot breathe.
little difficult in breathing.
Mhm.
Then system senses that please start producing erythropoietin because his oxygen levels are dropping.
Mhm.
So blood cells synthesis will be more.
Mhm.
Erythropoietin induce the RBC synthesis.
Mhm.
So whatever the amount of RBC, more and more RBC, try to grab the oxygen.
Mhm.
See, oxygen availability is less, but more people are there to share that oxygen. System thinks that oxygen is less, so let us capture more oxygen. So that is what kidney is sensitive, very sensitive to the low oxygen level. So we are understanding why this hypoxia causes any defects in proteinuria, because people who high altitude they have proteinuria than people who live at sea level.
Yeah.
Then I said growth hormone. I said advanced glycation and products. So we took at least half dozen of the agent that causes the renotoxicity. For example, glucose,
Mhm.
advanced glycation and products, hypoxia, then growth hormone, silica, and pesticides. So
Okay.
these are things we took it. Then we are seeing where we are exposed our podocytes or animals to these conditions. Then we study what is the difference in the kidney architecture, glomerular function when compared with healthy people versus uh, treated with these agents respectively.
So till now it is not understood at cellular or molecular level how hypoxia condition is affecting the kidney, and then that eventually is affecting human health. So once you understand it, you will know the reasons and you will maybe develop some drugs or some therapies so that people living in that condition can avoid this damage
Uh,
to kidney because of hypoxia condition.
Very good, excellent. You asked very relevant question.
Yeah.
Um, uh, it has a it is like two edges of the what you call your question. Your question was actually, can we offer any therapeutic approaches for the people high altitude? Okay. See, people who are living for ages generation high altitude, their levels also high, but they adapted. For
They
example,
adapted.
South American population who are living in the mountains, Brazilians, and uh, but what exactly physiology, that's very complex. For example, Peter Radcliffe, who is a British scientist who received a Nobel Prize in 2019, I guess, for this, he discovered, he identified a molecule that can inhibit the what do you call, that can induce the hyphon alpha, that can simulate the hypoxic condition. For that purpose, see, that time he or their group doesn't know that adverse effects of hypoxia. See, as I just mentioned, if you can induce hyphon alpha can induce erythropoietin so that RBC synthesis can happen. That is a direct effect of hypoxia. See, whenever you get the hypoxic condition, RBC synthesis will happen. So people with this chronic kidney disease, they have anemia. People with anemia, they can get chronic kidney disease, or chronic kidney disease people have anemia, vice versa. So he's a doctor actually. What he proposed, people with anemia, because low iron levels, if I try to give the drug that can induce the hypoxic condition, he can cure it. That was that for that approach, he got 1/3 of Nobel Prize actually. But he referred my paper, which we published later on the same year where he got Nobel Prize. He wrote published it in very good review in the Nature Nephrology. He wrote it that in the recent times, Nakluri, who is my PhD student, first PhD student, Nakluri et al. Showed that adverse effects of the induction of hypoxia in a glomerular level. Actually, he injected to that whole animal level. He saw that hemoglobin levels are catch up. That was the doctor's expression. But when we micro dissected the kidney function, we are trying to show that besides, though you claimed that one is a positive side of the hyphon alpha induction of the simulation of hypoxia, but we have adverse effects of simulation of hypoxia. That's what we showed, and we continue to show the adverse effect actually in people with, for example, stroke. The person who gets the stroke, his physiology responds ahead of the time. Proteinuria predicts the stroke risk. So what we did was we observed the kidney function, especially podocyte biology, in people, what do you call, proteinuric condition in the patients, and also simulated the stroke condition in a rat. It is called MCAO, middle cerebral artery occlusion. It's a model to simulate the stroke. There we found that in the stroke-induced animal, we could see the proteinuria and damage to our podocytes, glomerular filtration barrier. So hypoxia is it's a cliche. Hypoxia is though it appears that it's at the organism level it helping, but at a ground level it's not. It's also other effect. Okay. So now, what you can treat, how you can treat hypoxia, just to maintain normal level of oxygen, not super level of oxygen. Again, high oxygen is dangerous.
Yeah.
So you have to maintain oxygen level. That's why people who lives in people who go to the mountaineering, they suddenly cannot go to the high altitude suddenly. Step by step, that is called acclimatization. Our military people, after reaching some height, they take rest. After reaching further height, they take rest. Okay. Like that, and try to avoid the frequent mountaineering. That's the best thing actually. As of now, blocking, suggesting how to overcome hypoxia, like you have to oxygenate your body at normal level. That's the best thing as of now. See, we are telling the people that don't expose hypoxia. That itself is a good information. See, if you have a choice, don't go to frequent flyer. Actually, frequent flyers get this mild proteinuric condition.
Okay.
And to continue one thing, a recent discovery from our lab, hypoxic condition is stimulating calcium influx into the kidney cells, calcium influx. Hypoxia induces some kind of transient calcium receptors. So we are showing that the calcium receptors are blocking the calcium channels, helping these people. Our study, continuation to our study, we used calcium blockers to save that animal from the proteinuric condition and hypoxic condition. We continuously performing that study, and we are confident that calcium blockers are actually good. So even stroke patients also advised to give the calcium blockers.
So how that actually, calcium blockers?
Calcium blockers block the when all these hypoxic conditions, according to our research, hypoxia conditions induce one transcription factor that is called HIF1 alpha. HIF1 alpha stimulates the transcriptionally activate the transient calcium channels. So calcium is very high extracellularly and very low in cell. What happens as soon as your transporters or calcium channels are induced, more calcium try to gets into the cell. So excess calcium alters the inside homeostasis of the cell. For example, excess calcium, it goes to the endoplasmic reticulum. From there, it goes to the mitochondria and toxic to the mitochondrial function.
Okay.
So cells undergo immense stress because of excess calcium. As soon as excess calcium gets in, cell also try to push out the calcium. But anyhow, this calcium, store operated calcium and this excess intracellular transient calcium, all things are, they're changing the physiology of the healthy cell, and that's how it causes the mitophagy. One of the adverse events of the excess calcium is mitophagy. So we are currently we published till half actually. Excess hypoxia causes calcium influx. We showed, we proved a couple of times. Other people are also supporting, publishing their papers in support of our data, and we are trying to stop that. Mitophagy. Let's see whether we can block the calcium channels and can prevent or it can partially reverse the adverse effects of the hypoxia on podocytes. That's what we're trying to pursue.
Okay, but I'm still not able to link it. So excess calcium in the cell, how is it going to help the animal with stroke, or is it not?
It is adverse effect actually.
It is adverse. It is
effect.
Making the condition worse.
Yeah. When clot occurs in your brain, it is senses hypoxia. Suppose patients with stroke, some small clot appears, it won't allow the oxygen exchange. So hypoxia arises. So excess clots in the brain, they induce a systemic hypoxic condition. So kidneys will respond for that.
Okay, so what this is suggesting is that if someone is having stroke, then you should make sure that in the cells, excess of calcium should not be there, which will lead to hypoxia, which will make the condition even worse.
I'll put other way.
Okay.
If patients are are animal models, what we studied, if they get the stroke, stroke is nothing but a clot of the bloods actually.
Okay.
So there is a anoxic condition, insufficient supply of oxygen to cerebral region.
Okay.
So brain, this carotid arteries, the system that senses oxygen will sense that there is less oxygen going to the brain.
Yeah.
So brain continuously need oxygen. Why? Because oxygen is a final terminal electron acceptor of mitochondrial electron transport chain. So
ATP generation.
Yeah, my job of mitochondria is to give ATP, so that ATP function will be mitochondrial function will be altered. So system senses that you need excess calcium excessive oxygen to send it to the brain. So once there is anoxic condition in the brain, it will sense it by the carotid artery, and it will tell the kidney, please do something, please induce erythropoietin to help the brain.
Okay.
This is multi-organ connection.
So how is that going to happen?
Okay, now see, the brain, whatever the signal from the brain, it received by the kidney over responds by secreting more erythropoietin. So during this problem, hypoxic condition prevails in the kidney also. Whole body of us will sense hypoxia. So HIF1 alpha, which is a hypoxia mediator, HIF1 alpha is the one that is actually induces erythropoietin. So when you have low oxygen, your RBC has to be more blood cells will synthesis. That is called erythropoiesis. Who is helping erythropoiesis? HIF1 alpha.
Yeah.
So HIF1 alpha, one side is helping by inducing RBC synthesis. That is called erythropoiesis. Other side, it is also inducing the calcium influx into the cells.
So now I understand the hypoxic condition will lead to more erythrocytes in the blood, and they will carry then oxygen to deal with that hypoxic condition.
To deal with hypoxia.
Yeah.
But side effects is you have excess calcium, because see, when you switch on the light, you may think that it's not a polarized light, it is a non-polarized light. That means light goes to all directions. Suppose you want to have a light in only one portion, it is not possible, and you cannot spot light on something. So when you induce transcription factors in your body, you won't expect only one function will offer. Because of that one, there's a radiating effects are there, and HIF1 alpha induces more than 200 targets of HIF1 alpha are there, like a VEGF, vesicular endothelial growth factor, abnormal proliferation of endothelial cells, I just said HIF1 alpha induces several transcription factors that also induces MMPs, like matrix metalloproteases. One another adverse effect of the HIF1 alpha is that it is inducing the zinc finger E-box binding protein 2. So this zinc finger E-box binding protein 2, it is in short form it is known as JEB2. It has adverse effects on epithelial cells. Whenever JEB2 levels upregulate, it will suppress the epithelial features of a cell. For example, ecadherin, that is contributes for epithelial nature. See, podocytes are epithelial in nature. They're attached to the substratum, basement membrane, and offer the protection to the blood vessels. But when HIF1 alpha induces JEB2 in podocytes, JEB2 suppresses the ecadherin. So cells cannot attach to the bottom basement membrane. So podocytes gains the mesenchymal nature, that means floating nature. So slowly these podocytes will detach and they appear in the urine. They'll detach into the urinary space.
Okay.
So this phenomena we observed in animal models where we simulated the hypoxia, and also in diabetic nephropathy models, podocytes appear in the urine. Even in the patients with adverse diabetic nephropathy, we can isolate the podocyte exosomes and podocyte RNA, podocyte markers. So these are the adverse effects of hypoxia.
Okay. Yes, sir. I got.
Maybe I'm overdosing with a lot of scientific
no.
jargon.
Now all the dots connected. Why this is.
And if you permit me, I would like to share something.
Please.
This curious case of our cells. Very curious case. What I'm saying here, in embryo, our maternal and paternal cells will fuse and they will give one single cell. It's a diploid cell. This cell divides up to 16 cells. Then there is a process called cleavage. What happens during stage? 3 layers like ectoderm, endoderm, mesoderm. They separate out. In ectoderm, you have a nervous cells and skin, and mesoderm some organs, and endoderm even further organs. So what happened during this stage? Cells will migrate and settle as those organs. Cleavage, coelom kind of organs. Completely these organs will form. This process, the cells, they migrate and settle as epithelial cells. So I'll reiterate the fact. Mesenchymal cells, they settle as epithelial cells during organogenesis, during the development.
Okay.
But in the disease condition, epithelial cells try to become mesenchymal cells. For example, I'll tell you just now, in case of nephropathy, our podocytes are attaining though actually they're epithelial cells. Epithelial means they provide the coverage to the fixed place. These epithelial cells are gaining the mesenchymal nature. They become motile. In cancer condition, actually majority of and a significant portion of the cells are epithelial cells. They get they transform into mesenchymal cells. That is why epithelial cells detach, undergo metastasis. They become mesenchymal, undergo metastasis. They go somewhere place and again settle, and again they try to become a epithelial, and after some time again mesenchymal. So they're propagating their cancerous nature. That is called metastasis. In embryogenesis or organogenesis, what happening in the development? Mesenchymal cells becoming epithelial cells. That is called temporary phase transition, mesenchymal epithelial transition. But in disease adult, what happening? Epithelial cells becoming mesenchymal transition, EMT. So MET happening in embryogenesis, EMT is happening in a disease. But interesting thing is that though they are reciprocal phenomena, but what interesting thing what we found was there is some selected pathways that are very active and contributing for the MET nature in the embryogenesis. Same signaling events they're acting in adult cells. They're causing EMT. So, for example, I'll tell Notch pathway. We published this work a long time back in 2019 in a very renowned journals, and after that people are showing same pathway, and we also further studied the same pathway, and we are reproving that our hypothesis is still correct. So here is what happening. There is one pathway called Notch signaling. It's very essential for kidney development, organogenesis, and migration of those cells is called MET. Same pathway is active during hypoxia, during excess growth hormone conditions in acromegaly and diabetic nephropathy is very active, and it is also seems to be active in excess glucose stimulation conditions, which is showed recently by other people. So context-wise, this pathway, these signaling events, switching the either EMT or MET. Context-wise, in adult, they're triggering the EMT, migration of epithelial cells and loss of those cells, and in the embryogenesis, organogenesis, mesenchymal epithelial cells. Very interesting.
Okay.
This is, as I said, curious case really, and our focus has been on these developmental pathways, how these developmental origins of adult diabetic kidney disease. That is actually theme of my lab. So developmental origins of diabetic kidney disease. That's actually in nutshell what I would like to tell. So we're studying different transcription factors. For example, just I mentioned zinc finger E-box binding protein 2. Also another transcription factor is known as WT1, Wilms tumor 1. So how these transcription factors are context-wise, and Notch1, NICD. Notch is another transcription factor. NICD1, Notch intracellular domain 1. That is also another transcription factors. How these transcription factors are, function in a differently in the context-wise. The development they're helping, and in disease they're devastating. So here we are trying to design inhibitors for preventing the Notch activation. So there is drugs that failed in the clinical trials. We used those drugs in our model system. If we inhibit the Notch activation, we successfully prevented the loss of podocytes.
Oh.
And also recent study, couple of weeks back, we published in a renowned journals where we showed that podocytes in disease condition acting as inflammatory cell. These podocytes, like this is wonderful
Source
thing what
of
we observe.
inflammation.
Pardon?
Source of inflammation.
So yeah, it is actually inducing TNF alpha and TGF beta. Actually, people very long time they hypothesize that TNF alpha and TGF beta involved in the glomerular kidney diseases, but precisely the precise role where they produce, which cell type is producing this pro-inflammatory cytokines is not known. But in diabetic settings, podocytes themselves, they're transforming to a partially inflammatory cell and inducing these molecules, the secreting out and inviting the inflammatory macrophages.
Oh.
Actually, they're converting the monocytes into the macrophages. Monocytes are a kind of chaperoning cells, police kind of cells. They do policing at the injury, but if they convert it, transform into macrophages, they're kind of pro thieves actually, not a good cells in macrophages. So podocyte secretome, what are the inflammatory molecules secreted by the podocyte, they're transforming monocytes to macrophages.
And then those macrophages are damaging.
Accumulation of macrophages, it further progresses the inflammatory response. So we're questioning the age-old paradigm that a cell remain as a same cell type throughout its lifetime. It is not like that. Cell, it is based upon the poking or inflammatory inflammatory or disease stimuli. You call them as a cue, disease cue. Whatever based upon the signals that is coming in a different pathophysiological condition, the cells are attaining different phenotypes. If you permit me, I would like to tell about another curious case of serendipitous discovery from our group. These all are terminally differentiated cells. They never should divide, as I told before. For example, nerve cell is a terminally differentiated cell. Podocyte is a terminally differentiated cell. So what happening with these cell? For example, the cell always rest at a G0 stage. You call them as a quiescent stage or resting stage.
Yeah.
What we noticed in diabetic nephropathy, these podocytes are unnecessarily poked by these external stimuli. You call it as a mitogens or whatever it is. For example, growth hormone or excess glucose conditions. The G0 resting cell was forced to undergo cell division. Yeah, we showed by multiple ways, by FACS, by using pCNA, by duplication of the genetic material. So is a big cell inside the nucleus. Chromosomes are getting doubled and further 2x the chromosomes become doubled, they become 4x and all they become keep on dividing, multi-copy. Simultaneously, one would expect that cytokinesis should happen, but cytokinesis is not happening.
Okay.
Though there is a cytokinesis is not happening, but karyokinesis is happening. Nuclear division is happening, cytokinesis is not happening. So within a cell, you could see more than two nuclei are connecting to each other. They're not completely separated out. The spindle formation is not pulling the chromosome, so-called chromosomal material to into two daughter cells, and cytokinesis is not happening.
Okay.
So yeah, there is a
Multiple nucleus is in
yeah
the
more
cells.
than two nucleus are sitting at cell become bulged.
Okay. And that is
So
impairing the function of the kidney.
kidney. So what happened? The gloom is becoming bigger.
Yeah.
The podocyte is becoming bigger. See, for example, there is limited space is there, and we both are sat next to each other. If I become obese, I push you or I disturb the architecture of whatever our seating system. So system is already
Pretty
existing in a
dense.
proper architecture.
Okay.
Obesity of hypertrophy of this podocyte is causing the impairment of kidney function. That was our recent discovery. One beautiful thing is that another people first time we proposed this, where we demonstrated that in diabetes settings it is happening, and another Chinese person continued our study. He showed that a connection to this mitotic catastrophe, it is called mitotic catastrophe. What happened when more genetic material, more and more genetic material is accumulated without cell division? Cell want to die because it cannot handle that much amount of the nuclear material accumulation and mutations in the because of that. Cell automatically undergoes suicide. That is called mitotic catastrophe. So this is what we showed, and relation between mitotic catastrophe and one long non-code RNA was proved by recently, like 3 months back. The person, a Chinese guy, demonstrated that there is a connection between long non-code RNA. So fortunately, we reviewed that paper and we wrote a beautiful editorial on that one. So it's a good thing that we're trying at least to show that what are the toxicity, what are the adverse effects that are happening in a diabetic settings, and how podocytes are trying to cope up with or trying to become susceptible for them. So it is better to prevent podocytes at early stages of diabetic situation, because podocytes damage is a very early event in diabetic nephropathy progression. So we have to maintain this podocyte number, podocyte quality is very important. So we're trying to design a targeted drugs to prevent the podocyte damage. Yeah, because of some reason we are unable to disclose, because we are trying to go for intellectual patenting, and this thing. All students in my lab, day and night, day out, we're trying to first of all, we love to protect the kidneys, and we try to offer at least some therapeutic solution to the mankind to prevent the podocyte injury and podocyte loss. I'm dreaming we come up with some strategic therapeutic options very soon. That was in my optimistic approach. Yeah, and I mean it for that.
Yes. Thank you very much, sir.
Yeah,
Some
thank you so
questions
for
I had,
for
and it was really
it's
interesting to know.
no one
what-
asked such intense questions actually before you.
Okay, podocytes. I mean, I knew about diabetes. I mean, whatever I studied in bachelor's and master's, and right now also, but to get it know at such a deep level, at the level of podocytes, because I mean, yes, there is a high glucose level and everything, but how that actually is connecting to the physiology that we see with the diabetes people, that I understood today. Like actually podocytes is getting damaged, and then there is ketogluria and the excess of proteins in the urine, and then we lose important proteins, and that will impair the function. So thank you very much, sir, for
Yeah,
explaining.
I'm very happy. Actually, for me also, to explain. Uh, Usually, we tend to explain to the people who are in a kidney biology and they understand it in our scientific jargon. Though you are from scientific background, but this much detailing of what do you call kidney, and thanks for giving me the opportunity. It's absolute pleasure for me to have a conversation with you. Huh?
Thank you, sir.
Yeah,
Yeah.
thank you.